Okay, while you all finish the attendance quiz, a few more comments about the test. So there was a wide range. Some people got perfect or near perfect, and with the bonus marks you would have gotten a little better than perfect. But some people definitely have room to improve, let's say. Luckily, the first test is only 15% of the grade, so if you didn't do that well, don't take it as a sign of doom. Take it as a sign that you need to study more. Please take advantage of the resources on Blackboard. So one that I wouldn't have been able to show on the first day, oh no, it's not here at all. Okay, after class, I will add a link to the lecture playlist. That's too bad. So there'll be a link there to, and actually, even in general, if you just Google David Simpson Psych, you'll find my YouTube channel, and then there should be a playlist there for some. I did make an announcement, okay, but I was going to add it as a separate thing too. So in any case, it's at least on the announcements. You can Google it, and at the end of class, it will be here as well. So take advantage of that. You can rewatch the lectures there. The most important thing, as I say, is the key terms list. So if you didn't do as well, before the test, quiz yourself on those terms. And I'll add, you should try doing that before test review day. Only a handful of people came to test review day, but if you didn't do as well on the test, that's a great chance to come and ask any outstanding questions that you have. So that'll be the Friday before every test. So next one's already coming up in not that long, so that's next Friday. Because only 2 chapters before the next test. And of course you can ask questions here as well. Okay, any questions about the test? Okay, so today we're talking about the brain. So one of the more bio-heavy chapters in 1100. Other chapters will make some reference to it, but if you didn't take Bio 12 or anything like that, may be harder, but I'll be sure to cover it slowly and step by step. So if you didn't take Bio 12, you'll still be able to get 100%. Actually, I didn't take Bio 12 for whatever that's worth. I'm going to start with some common slogans that you'll hear if you speak to psychologists and neuroscientists. These slogans are emblematic of how they think about the brain. One slogan you'll often hear is, the mind is what the brain does. This is a rejection of the view that we saw in chapter 1, namely dualism. Psychologists typically operate with the assumption that there's not an extra magical part of the human person. It's all just biological. So primarily the brain, other parts of the body contribute to things like emotions, but the brain is where the action is. Another slogan you'll hear, which is more to the point, everything psychological is simultaneously biological. If something changes in the mind, there is always a corresponding change at the biological level of analysis. So that's kind of the standard view. Now, as a caveat, in the chapter on consciousness, we will see a challenge to this view. This view being materialism, the view that our mental lives depend on the brain. Consciousness poses an interesting problem for this view, and we'll see that in more detail in the consciousness chapter. But let's start with some facts about the brain. So, the 3 pounds of meat between your ears, this is what it looks like. The brain has around 85 billion neurons, which we'll be talking a lot about. And this is harder to estimate, but something like 100 trillion connections between those 2 neurons. It is the most complicated thing that we know of in the universe. Even though it's only 2% of your body weight, it consumes about 20% of your calories. So it's a very greedy, very expensive organ. And as I say, psychologists now believe that this is the source of mental life. And I'm going to show a video from Paul Bloom talking about this. So, talking about one line, so there's different lines of evidence for thinking that the mind depends on the brain. The one that he's going to make reference to is different correlational studies. So you can get people to engage in various psychological tasks, and there's always a corresponding change in the brain. So yes, that just underlines the point that it's taken for granted today that all of our different psychological functions depend on brain activity. In that video, he's making reference to correlational studies, but we also have causal evidence. So, as you should all know by now, correlation does not imply causation. In terms of causal evidence, we'll see various cases in this chapter related to brain damage. So for any psychological function that you care to name, you can point to cases often where if a given part of the brain is damaged, you will no longer be able to perform that psychological function. So whether it's generating memories, personality, different types of emotions, those can be taken away by damaging parts of the brain. And here's a simpler case that all of you are familiar with: sleep. Every night, what happens? Activity in the brain goes down. What happens to your mental life? What happens to your conscious experience? Well, at least during the dreamless parts of sleep, it goes away, right? No conscious experience left. And you can make a similar point about the effects of drugs, right? Drugs that impact the brain's chemistry and activity change your mental life. They change your conscious experience. So, now we'll start working our way through different findings from neuroscience, and we're going to start zoomed in. We're going to start with the basic building blocks of the brain. I'm referring, of course, to neurons. So, neurons are the cells that send signals to each other. And this is happening billions of times every second. Neurons are constantly sending signals to each other, and this is what enables us to think and feel and process information. There are other cells in the brain, like glial cells. Glial cells are sort of the helper cells. They're not involved in thought in any interesting way, so the neurons are where the action is. So the middle of a neuron is called the cell body. This is the boss of the neuron, if you like. And by the way, I will be speaking that way throughout this chapter. It's common when teaching introductory neuroscience to personify different parts of the brain, to speak of them wanting things or aiming at something. This is a bit of poetic license. Obviously, they don't literally, think or feel in the sense that we do, but it can be a helpful first approximation. So the cell body is, as I say, the boss of the neuron. The appendage going out from the cell body, the big one, is the axon. The axon is what sends signals to other neurons. The appendages going out from the back are called, let me see if I can better, are called the dendrites. The dendrites receive signals from other neurons. Now, the neuron will typically have one axon, but multiple dendrites. You may think, how does that work with the connections? The way it works is each axon will have multiple terminal branches or terminal buttons. So therefore, one axon can send signals to multiple neurons. Another very important part of a neuron is this. Well, not really a part. It's a gap. Is the synapse. When one neuron sends a signal to another neuron, the axon and dendrites do not touch. There's always a little space between them, and chemicals will be sent across that space. Up to the end of the axon, the signal is transmitted via electric charge, but the charge does not cross the gap or synapse. It causes chemicals to be sent across that gap. And as I say, the term for that gap is the synapse. The axon is covered with tubes of fatty tissue, and these are called the myelin sheath. Their function is to increase the speed of neural signals. So the charge moves up the axon, the myelin sheath ensures that the charge moves more quickly. How many of you have heard of multiple sclerosis, the condition? So it's a medical condition, and it's caused by the body's immune system attacking the myelin sheath. And this has various effects for movement and thinking, right? Because it makes it more difficult for the brain to transmit signals, or for the neurons in the brain to transmit signals. By the way, so I mentioned in passing the glial cells. So the glial cells are like the helper cells. There's one type of glial cell that constitutes the myelin sheaths. That's one example of the job of certain glial cells. There are other glial cells that clean waste, various other things. The book calls them the support cells. Okay, there are different types of neurons. There's different ways of classifying them. There's only one classification I want you to worry about. You can break this down further, but this is good enough for an intro psych course. The three types you need to know are sensory neurons, interneurons, and motor neurons. Sensory neurons, as you may be able to guess, are connected with the sense organs. So the skin, the cochlea inside the ear, the eyes. So take the eyes, we'll be seeing in a later chapter. In that case, the sensory neurons are called rods and cones. They are stimulated by light, and when they are, they send signals to the brain. So that's the function of the sensory neurons. They are stimulated by things in the environment, and then they send signals to the brain. Motor neurons send commands to the body to move and do other things. So, my motor cortex sends signals to my hand, making it do this. It's motor neurons that send those signals. All the other neurons in the brain are interneurons. So the vast majority of the neurons in the brain will fall in that middle category. So all of the processing of those signals is accomplished by the interneurons. So just to take a silly example, if one of you gets tired of the lecture and throws something at me, so the light will bounce off the object to my eyes, the sensory neurons will send signals to my brain. The interneurons will process it, and it's a red tomato, it's moving this fast. And then put together a plan of, hey, you should put your hands up to avoid it, or to slap it out of the way. And then the motor neurons will send the signals to my hands. simple case to remember what they do. Okay, any questions about anything I've said so far? Okay, I'm going to try and go slow through the next section, because the next section is what gave me the biggest headache when I first took intro psych. And that's how neurons send signals. So, start with the basics. Neurons, when they are at resting potential, are negatively charged. So if they're not doing anything, they're just chilling, being neurons, they have a charge of around minus 70 millivolts. So let's say this is 0, they're at around minus 70. They are surrounded by positively charged sodium ions. So the sodium ions stay on the outside when the neuron is at rest. Other neurons will try to depolarize the neuron. So they're saying, hey, come on, become positive. Some neurons will make it more negative, but if it's an excitatory neuron, which we'll talk about later, they will try to make it more positive. At around minus 50 millivolts, there's a magic line called the threshold. If the neuron goes above that magic line, something special happens. So it goes up above this. Once it goes above that line, something happens. All of the sodium channels open, and the positively charged sodium is attracted in. So you should remember from high school that negative charges attract positive charges. So the sodium rushes in because it's gone above the threshold, and then it goes up. The positive charge is then what moves up the axon. So that event is called an action potential. Is that the sodium comes in, positive charge, and the charge moves up the axon. And to foreshadow what comes next, when the charge goes to the end, it causes chemicals called neurotransmitters to be sent across that gap. Is everybody with me so far? Now, once it becomes positive, the neuron wants to go back to being negative so it can send a signal again. This is kind of the confusing part. As we'll see throughout this chapter, evolution did not design the brain to be easy to understand. So positive sodium is what rushes in, but inside the neuron already, before that happens, it keeps some positive potassium kicking around, so it can kick it out later. So it has the potassium in there already, and it kicks it out to get it back down to negative. So the positive sodium is what rushes in to cause a charge to move up the axon, but it kicks out the potassium to go back down to negative. Does that make sense? You don't have to know this for the test, but you might be thinking, well, wait, if it keeps letting sodium in and kicking potassium out, won't it like run out of potassium? And for whatever it's worth, all neurons all the time have sodium-potassium pumps that kick sodium out and bring potassium in for later. That's how it keeps the potassium on the outside most of the time and the potassium in. But don't worry about that for the test. What you should know for the test is it's negative by default, but once it goes above the magic line, It opens up the sodium channels, and the sodium comes in, leading to this positive charge, which is what moves down the axon. And that to go back to negative, it kicks out the potassium that it has kicking around. So I've tried to go slow to avoid headaches, but any outstanding questions about an action potential? So these are happening all the time. Individual neurons can, in some cases, send dozens to hundreds per second. And you have 85 billion neurons in your head, so pretty big number. Kind of fun to think that this process right now is what's enabling you to understand what I'm saying. This should also be humbling, I think, because we like to think that we're very special, but when you zoom in to individual neurons, it's just basic chemistry. It's just negative charges attracting positive charges. That's really all the brain is. Something very important you have to know about action potentials is they are, as the book says, all or none. There is no such thing as a weak action potential or a strong action potential. It doesn't matter how quickly it goes up to and past the threshold. Once it goes above that threshold, a signal will be sent. If you like, you can think of it like a gun. If you pull the trigger, the gun either fires or does not fire. If you pull the trigger fast, it doesn't make any difference. As an aside, I actually knew someone who got shot in the head. I used to work a concrete type job, and they'll hire anyone. The guy had some Hells Angels affiliations, and things happen when you have said affiliations. And he told me about his incident within 5 minutes of meeting him. He was a bit of a character. I mention this also because it's one of the rare cases where my psych knowledge actually came in handy, because I had heard about him in advance. My brother had already met him, and apparently he had a kind of out-of-body experience after being shot in the head, and as a result converted. And when I heard that, I predicted that he'd been shot on the right side and that it had been here, because there's an area at the kind of back of the temporal lobe, the temporoparietal junction. When there's dysfunction there, often people report having an out-of-body experience. And when I met him, again, within 5 minutes of meeting him, so, I got shot in the head right here. And I was like, yes, psych knowledge came in handy. Anyways, the neuron is like a gun. It either fires or does not fire. I mentioned in chapter 1 that many people try to draw an analogy between computers and the brain. This is another place where you could draw an analogy, because computers are like that, right? It's binary code, either 1 or 0, which at the small scale means electricity either runs through or doesn't, and the brain works like that as well. One other term, when the neuron is going down, because it's kicking out that extra potassium, this is called the refractory period. Technically it goes down a bit lower first. So during that period of time, the neuron cannot send another signal. It can't initiate another action potential. So the term for that is the refractory period. If it helps, I'm just going to say something general. In general with this chapter, if you can think of funny or even rude analogies or memory aids, that will be helpful. A lot of the stuff is pretty dry. For whatever it's worth, refractory period is also the term for the period of time in which after a man orgasms, he cannot have another orgasm. If that helps, you can use that. So that's the kind of electric part of it. So charge moves up the axon. Now we're going to look at what happens here at the synapse. So we're just going to zoom in to that part. Any questions before I erase all this? So the charge moves to the end, and the end of the axon sends little chemicals across to the dendrite of the other neuron. The little chemicals that move across the synapse are called neurotransmitters. You've probably heard of some of these: dopamine, serotonin, those are all neurotransmitters. And then they bind with receptors in the next neuron. Not all of the neurotransmitters will bind with a receptor. So let's say these ones don't. What the prior neuron will do is it will take some of these back. If they don't bind with any receptors in time, no one swiped right for them. Nature is mean. It takes some of them back to be recycled. That is called reuptake. Many psychoactive drugs change brain chemistry by acting on this process. So there are many drugs, for example, that prevent reuptake, which means that the neurons have more time in the synapse to bind with the next neuron. So it's a kind of a double negative. If you decrease reuptake, more of that neurotransmitter gets transmitted across. Okay, so now we'll start talking about specific neurotransmitters. One broad distinction is the distinction between excitatory neurotransmitters and inhibitory neurotransmitters. Excitatory neurotransmitters increase the chance that the next neuron will send a signal, bring it closer to that threshold. So they're like, come on, man, send a signal. Inhibitory neurotransmitters decrease the probability that the next neuron will send a signal. So they're like, hey, no more signals today. Some neurons can do both. There are a few that are exclusively one or the other. We'll see some of that as we go through. You don't have to know for each one whether they're inhibitory, excitatory, or both, but for some of them it's important, and I'll make a point of saying so, and it is important. Unfortunately, both for neurotransmitters and for brain areas, there's a lot of overlap. I already mentioned that evolution did not design the brain to be easy to understand. Would have been nice if each one had one job, but alas, that's not how it works. So the first one is acetylcholine, involved in a number of different functions. It's involved in voluntary behavior, also involved in learning and memory. Part of the reason we know that is people with Alzheimer's disease, which is a memory problem, have lower levels of acetylcholine. I don't think I mentioned this to my Tuesday class, but it's also increased by smoking cigarettes. Cigarettes do improve your memory while using them. Probably not worth the lung cancer, but it does act on acetylcholine. The next one is dopamine. Dopamine, like acetylcholine, is also involved in voluntary behavior. Probably its most well-known function is it's involved in the experience of pleasure or reward. As we'll see later, the nucleus accumbens is what sends dopamine throughout the brain when you experience pleasure. Some studies find that high levels of dopamine are associated with schizophrenia. Somewhat controversial, but some studies find that. Low levels of dopamine are associated with Parkinson's disease. Parkinson's disease, of course, is a motor problem associated with difficulty moving. So that makes sense because dopamine is involved in motor behavior. Next is glutamate. Glutamate is the brain's main excitatory neurotransmitter. So if glutamate is sent from one neuron to the other, it increases the chance that the next one will send a signal. It's important for forming memories. When two neurons, if the connection is repeatedly stimulated, so they repeatedly send signals, the next neuron becomes more sensitive to those signals, which means the connection is stronger. And it's the hippocampus, which we'll talk about later, that accomplishes this. It repeatedly stimulates connections between neurons via glutamate. So it's very important for memory. If you have too much glutamate sent throughout the brain, you get seizures. You get kind of a brainstorm. Not a good kind of brainstorm, but excessive sending of signals, and the person loses control. So glutamate is the main excitatory neurotransmitter. The main inhibitory neurotransmitter is GABA. You don't have to know what it stands for. GABA is fine. So if GABA is sent from one neuron to the other, it tells the next neuron, so to speak, to not send a signal. So if more GABA is released, there's less brain activity. So one drug that I'm sure most of you are fond of, your first year students, is alcohol. Alcohol increases GABA transmission. It does so in the frontal lobes first, which makes sense, right? A lot of foolish decisions made while under the influence of alcohol. I'll be telling some stories about that in a later chapter. Not about myself, I was a good boy. But if you take too much, it can affect other parts of the brain. If it goes down and affects the medulla, you can die. Because if you slow down the medulla, your heart stops beating and you stop breathing. So that's not great. The next one is norepinephrine. This is chemically similar to adrenaline, and it's involved in similar things. So an increase in norepinephrine transmission leads to more vigilance and alertness. The next one, serotonin, is involved in a number of different things. It has some connection with mood. That's probably what it's most well known for. I think that's sufficiently well established. However, one common claim you'll hear is that low levels of serotonin cause depression. That's probably false. The connection is probably not that simple. The reason many researchers assumed that is standard antidepressants, SSRIs, prevent the reuptake of serotonin, which, as I mentioned earlier, means more serotonin gets sent across if you stop reuptake. SSRIs do improve symptoms of depression slightly better than placebo, very slightly, but measurably better than placebo. And what people inferred from that is, low serotonin causes depression. But that's not a very valid argument. I mean, here's a silly analogy. If I give you heroin, you will be happier, at least for a while. That does not mean that low heroin is what caused your sadness. So probably it's not the case that low serotonin causes depression. There was a recent very large meta-analysis that looked at all the studies where they actually measure serotonin levels indirectly and look for a difference between depressed and non-depressed people, and they didn't find any difference. So there's some connection to mood, but it's not as simple as low serotonin equals depression. And And then the last neurotransmitter, don't be too sad that it's over, is endorphins. So endorphins comes from endogenous morphine. So this isn't literally what it is, but it means internally generated morphine. It's basically the brain's painkiller. So it's really the opposite of that. It's that morphine and other opiate-type drugs act on the endorphin system. So if you get injured and go into shock and don't feel the pain, that's the endorphin system at work. When you exercise, this exercise is basically damaging your body in little ways. Endorphins are released. This is sometimes called the runner's high. So those are some functions that endorphins serve. Okay, one of the most fun sections in the book. Any questions about neurotransmitters? As I mentioned, different drugs will act on the neurotransmitter system in different ways. We'll get into more detail in the later chapter, but for now, the broad distinction you should know is the distinction between agonists and antagonists. Agonists increase the transmission of a given neurotransmitter. So for example, cocaine is a dopamine agonist. More more dopamine gets sent across. Antagonists decrease how much of a given neurotransmitter is sent across. So antipsychotic drugs are dopamine antagonists, for example. Okay, now we'll zoom way out to the overall organization of the nervous system. So the first category, really easy. The central nervous system is just the brain and the spinal cord. That's it. So this is the command center. So the combination of the brain and the spinal cord is the central nervous system. All of the nerves that go out from the spinal cord to the rest of the body are a part of the peripheral nervous system. And this further breaks down into the somatic and the autonomic. So, autonomic sounds like automatic, and that's right. The autonomic system operates outside of conscious voluntary control. You can't choose to activate or deactivate your autonomic system, at least not directly. I mean, you could do something like breathe. If you breathe really heavy for like a minute, you will trigger changes here, but you can't just through conscious will change the autonomic system. The somatic system is the nerves that are associated with like touch perception, so like skin receptors in your skin, and the muscles that initiate movement. So if I choose to raise my hand, nerve cells going from the spine to the muscles in my arm, those are part of the somatic system. And if I touch this table and I feel the signals coming from my fingertips back up to my brain, the nerve cells associated with that are also part of the somatic system. So you're consciously aware of the action here, but you're not in control of this one. The final division is the division between the sympathetic system, also called the fight or flight system, and the parasympathetic, also called the rest and digest system. So the parasympathetic is business as usual. If you're calm and relaxed and nothing strange is going on, your body digests food, heals wounds, fights diseases, all that good stuff. If a tiger was to walk into the room, escaped from the zoo or something, sympathetic system lights up. What it does, you can ignore some of the details. What matters is it redirects energy away from things like digestion, immune functioning, etc., and tries to get all of the energy to the heart, lungs, and muscles. No point digesting lunch if you're about to be lunch, right? That's not a good use of energy. If you want to remember which is which, sympathetic also means nice, right? If we say someone's sympathetic, it means they're nice. I remembered, okay, evolution was nice to us, so it helps us run away from bears and stuff like that. If that helps, you can use that. This is why when, we've all known this since we were young, when people get really scared, sometimes they poop themselves. This is why, right? The body's like, we're not worried about that. That doesn't matter. All energy is going to the heart, lungs, and muscles. So in general, the way the system works when you're moving is the brain will send signals to the muscles, getting them to move. The exception to that is spinal reflexes. So the spine, you can think of it as being, not that smart, but it's smart enough to know when your body's being damaged. It's sort of designed to do that. So all of you at some point, I'm sure, have touched a really hot surface. Next time it happens, reflect on what it's like. Because what typically happens is your hand pulls away before you feel the pain. What's going on is the signal goes to the spine, and the spine can tell, this isn't going so well. We're going to abort mission and pull the hand away. In order to consciously experience the pain, the signal has to make its way to the brain, and I won't say which parts, but different parts of the brain will light up in response to the damage. But the spine is, can do enough processing to figure out that the body's being damaged and initiate a reflex. You can also trigger it by hitting the nerve cells, right? This is what they do in the doctor's office, right? Hit the nerve in the knee, you can initiate a reflex that way. Any questions about the divisions of the central nervous system, or of the nervous system? Okay, let's take a break a little early. So when we come back, we're going to talk about different parts of the brain, and there's more videos in this section. So see you all for that. So let's come back at 4:30. Okay, everyone, let's get started again. So we looked at the very small scale with the neurons, and then the very large scale with the division of the nervous system. Now we'll start getting into specific regions of the brain. So I guess the mediumly zoomed in. I'm going to start with some general principles. These are somewhat simplistic. They're not 100% true, but they're a good first approximation. They will help you memorize different parts of the brain. So this is what the brain looks like. The eyes would be around here, and then that kind of bumpy thing at the back would be around here. So general principles. The more to the outside and the more to the front a part of the brain is, the more evolutionarily recent it is, and therefore the more complicated it is, typically. The reverse is true. The more to the middle and the more to the back, the more towards the neck, a part of the brain is, the more evolutionarily ancient it is, and therefore the more rudimentary it is. So if you go to the outside frontal cortex, that's where you get the part of the brain that's most involved in complex reasoning, long-term planning, and the more distinctly human types of thinking. You go all the way to the middle and all the way to the back, you get to the medulla, which is basically just where the spinal cord gets thicker, and now it's the brain. What does the medulla do? Blood circulation, breathing, stuff like that. So you damage that, you die. So there are three broad areas in the brain. Now in humans, the middle one, the midbrain, is quite small. In other animals, the three are more equal in size, but we'll stick with the convention and talk about the three areas. The first is the hindbrain, which includes some substructures. So the hindbrain is around here. So this bumpy thing and this skinny thing in the middle, those are part of the hindbrain. So more rudimentary stuff. The first part is the medulla, which I just mentioned in passing. So basically, spinal cord gets thicker, now it's the brain. Very simple survival-related stuff. So keep your heart beating, keep the person breathing, etc. Next section is the pons. So just sitting right above the medulla. It's involved in sleep and arousal. Part of the reason it's involved in those two things is there's a structure that goes through that and then connects to the rest of the brain called the reticular formation. The reticular formation is sometimes called the brain's dimmer switch. So a dimmer switch is a light switch where you can make it brighter or more dim. The reticular formation has a similar function in the brain. It can increase or decrease arousal. If you damage the reticular formation, you will fall into a coma. So good to not damage that. So that's one thing that runs through the pons. Another thing that runs through the pons is the cranial nerves, which then connect with the face. So it also has a role in modulating facial expressions. This goofy looking thing here, which is visible even zoomed out, if you look at it, it kind of looks like a little second brain. And neuroscientists, being the very clever people that they are, named it the little brain, or cerebellum. So cerebellum literally just means little brain. The cerebellum is a fascinating part of the brain. It has more neurons than the entire rest of the brain combined. That may seem impossible. The reason for it is it has significantly fewer connections. The rest of the brain, especially the cortex on the outside, each neuron will have many connections with other neurons. The cerebellum is more feed-forward, one neuron to one neuron to one neuron. What does it do? It's involved in ensuring that well-practiced behaviors are done well. So if I was to remove your cerebellum, which some people, for various reasons, don't have one, your behavior, you'd still be able to do things like walk, type, etc., but they would be very clumsy, less well coordinated. So the cerebellum doesn't initiate decisions to move or do things, but it makes sure that they're well done. So that's the primary role of the cerebellum. So that's all the hindbrain, nothing too fancy there. Any questions about the different bits of the hindbrain? Okay, the next umbrella is the midbrain, which, as I say, in other animals, it's bigger. Only a few things you need to know about it. So the midbrain can do some very simple sensory processing, especially vision and hearing, and some simple guiding of kind of reflexive behaviors, or quick behaviors. So it's not involved in conscious sensory processing. So I look out, I'm having a visual conscious experience, I can see your faces. The midbrain is not doing that. Well, I should say the midbrain, I mean, not very surprising, but it's just above the hindbrain, so right around there. But if there's like a bright light over here, and I go, whoa, what's that? The midbrain is part of what facilitates that. Or if there's a loud bang, same kind of thing. Part of the reason we know that is a really spooky condition called blind sight, which sounds like an oxymoron, but when people have damage to the visual cortex, so this is the back of the brain around here, as we'll see later. If you have enough damage to that part of the brain, as far as your conscious experience is concerned, you won't see anything. If someone broke that in my head right now, it would be black, right? I would not see anything. But people with this condition have those more ancient, simpler parts of the brain that do visual processing still intact. So, you flash a light in the top corner of the screen, and you say, point to the light. And they say, this is mean, I don't like this study. And you tell them, just guess. And more than chance, they'll point in the right spot. Similarly, if they have to step over things in a crowded hallway, they will successfully do it, even if they report not being able to see anything on the ground. So structures like the midbrain can enable that, even if the person doesn't have conscious visual experience. So that's the midbrain and hindbrain. The forebrain is really where the action is. So in mammals such as ourselves, this is the biggest chunk of the brain. So all this is the forebrain. The forebrain can usefully be divided into two sets of systems. The book divides it up a little bit differently. There's some controversy about what to include in the limbic system. We'll go with this one. So another good first approximation that's not 100% true is that the cortex, which is the outside part, so the pink, bumpy looking stuff, that's the cerebral cortex, the outside of the forebrain. All these colored structures in the middle, those are part of the limbic system. And again, a good first approximation, the cortex is more involved in complex processing and reason, whereas the limbic system is more instinctive and associated with emotions. You'll learn complexities related to that if you move on, but that's a good rough sketch. So let's zoom in on specific parts. First part is the thalamus. The thalamus, which is in the limbic system, in some classifications anyways, is the relay center for sensory information. Of the 5 senses, 4 of them go to the thalamus first, and then get redirected to the cortex. So if light goes into the eyes, signal goes from the eyes to the thalamus first, and the thalamus goes, okay, your visual information, you go to this part of the cortex. If it comes from the ears, the thalamus will redirect it to a different part of the cortex. So that's the thalamus's job. The exception is smell. Smell cheats. Goes straight to be processed. Next part of the brain is the hypothalamus. They do not have similar functions. It's just called the hypothalamus because it's close to it. That's the only reason. Like spatially close. Anyone in 1200 remember the easy way to remember what the hypothalamus does? We would have talked about it already. Yeah, the 4 F's. So the hypothalamus is the boss of so one of them is hunger. So it's the boss of the hormone system, and therefore it's related to the sorts of things that hormones do. So the bad neuroscience joke is that the hypothalamus is associated with the 4 F's of survival: feeding, fleeing, fighting, and a 4th F. So if you're very sheltered, sexual functioning. So there you go. That makes it easier to remember. So right near the hypothalamus is something that's, I don't think in the key terms, but the pituitary gland. So the pituitary gland is near the hypothalamus, and the hypothalamus is its boss. So that's what it does. Okay. Next part is the hippocampus. Hippocampus comes from a word for seahorse. It's kind of shaped like a seahorse. And this part of the brain is important for generating new long-term memories. It doesn't store the memories, but it helps to generate them. What it does is strengthen connections in the cortex by sending glutamate. Part of the reason we know this is a famous brain damage patient named Henry Molaison. Before he died, he was referred to as HM. And we'll watch a video about Henry Molaison. So the short version is he had his hippocampus removed because of life-threatening seizures that were emanating from the hippocampus, and he lost the ability to form new long-term memories. As an aside, you'll see a female scientist who works with HM after the surgery. She's still alive. She works at McGill University and is still going strong at 105 years old. So, pretty impressive. Okay. So that should make it more memorable, no pun intended. So that's the function of the hippocampus. The next part of the brain is the amygdala. Technically there's two, one on each side. So the hippocampi kind of go out like this, and then at the end of each is the amygdala for each hemisphere. They're kind of almond-shaped. If you had to point to one part of the limbic system that's most associated with emotion, it would be the amygdala. So it lights up in various emotional experiences. The emotions for which the connection is most strongly established is fear and aggression, but it lights up during other types of emotional experiences as well. It's not an accident that it's at the end of the hippocampus. Part of the function of the amygdala is to prioritize different types of experiences for memory. Events that are more emotionally salient are more memorable. Here's a simple example. If you tried really hard to remember your experience of walking to class last week, probably not a lot comes up. You have to think pretty hard to get anything interesting. If someone punched you in the face as you walked to class, I dare say you'd probably remember it. Right, so you can think of the amygdala as like little alarm bells, like, hey, ding, 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 remember this. The next term is the basal ganglia. This is not one structure, but a set of structures in the middle of the brain. They have a number of different functions. Some parts function in a way analogous to the cerebellum. They help make sure that behaviors are well executed. So it's involved in voluntary movement. One part of the basal ganglia that's especially important is the nucleus accumbens, which I mentioned in passing. This has the job of sending dopamine everywhere during pleasurable experiences. And as a result, also important for forming addictions. All addictive drugs have an effect, either direct or indirect, on the nucleus accumbens. So food, drugs, sex, nucleus accumbens is important for all of those. So we already talked about the hypothalamus. The book then goes through some other regions that are relevant to the endocrine system or the hormone system. So there's the pituitary gland, for example, which the hypothalamus is the boss of. Okay, that's all I have to say about the limbic system. Any questions about those structures? Video. Okay. So now we'll move to the cerebral cortex. So this is the outside part of the brain. The term cortex means bark. That's the etymology of it. So you can think of like the bark of a tree. This is sort of like that. It's the outside layer. The cerebral cortex is, like most of the limbic system, is divided into two hemispheres, left and the right. We'll talk more about that later. The more important division for now is the division between the four lobes. So these are all good to know. So I'll start at the back with the occipital cortex. Most of the other lobes will have multiple functions. This one basically has one, and that's the early stages of conscious visual processing, so vision. So recall that signals get sent from the eyes to the thalamus first. The signals go then from there to the occipital cortex. So it will process things like shape, movement, color, those kind of early pieces of information. Since we rely heavily on vision, the parietal and temporal lobes help with vision a bit too, in ways that I'll describe later, but those first steps are done by the occipital cortex. If you want to remember, occipital is pretty close to the word optical. If that helps you remember, you can use that. So I'll do the temporal lobe next. The most important job that the temporal lobes have is auditory processing. So the primary auditory cortex, so hearing, is in the temporal lobes. Doesn't always work like this, but it happens to be near the ears, so that's kind of convenient. In the case of the occipital, it's the opposite. It's far away from the sense organs. The temporal lobes also help out with vision a bit. So the occipital lobe asks the temporal lobe, hey, help me out with this. And more specifically, figure out what this is. So for object identification, like, oh, that's a face, that's a ball, that's a rock, that processing is done largely by the temporal lobe. The parietal lobe up top, the way I remembered that in my first psych course was I thought it sounded like parent. So parents are on top, they tell you what to do and ruin your life, you know? So that's where the parietal lobe is. So it does a number of different things, including spatial reasoning. It's correlated with if someone has to do a spatial task, the parietal lobe tends to light up. The way in which it helps the occipital lobe is it does where is this? So the temporal lobe, as far as vision is concerned, is what is this? The parietal lobe is involved in processing where it is. So it also includes this dark red area, which is the somatosensory cortex, sometimes called the somatosensory strip. I'll come back to that after I do the frontal lobe. So the frontal lobe is the part of the brain where there's the largest size difference between us and other mammals. So our brains are bigger in general than chimpanzees, gorillas, and our other relatives, but that's especially true of the frontal lobes. Our frontal lobes are especially large. So as I mentioned in passing already, they're involved in things like long-term planning, complex reasoning, self-control, like controlling your emotions, etc. They are typically the last to finish developing. So they stay flexible for a longer period of time, presumably to take input from the environment. For example, social reasoning, right? It's very important to learn the social norms of one's community. Humans are and always have been a very social species, and being flexible and being able to learn those norms for a long period of time is important. You may have heard that male brains develop more slowly on average than female brains. This is true, especially of the frontal lobes. So if there are young men in your life that you feel are where you feel that's true, just give them time. I once asked my dad, because when I was a kid, I was already becoming a psychologist. I just didn't know it yet. And I had noticed a pattern. I was an empiricist. I was using my observation. I had noticed that young males were more likely to do violent, dangerous, foolish things. And so I just asked my dad, I was like, why is that? Why is it when there's some foolish, violent, or dangerous behavior, it's usually a male? And my dad said, that's because of testosterone. And I said, what's testosterone? And he wanted to explain it in a way that I could understand. So he said, basically it's a chemical that eats boys' brains. It's not exactly wrong, but not exactly right. So that's the frontal lobes. Now, the two remaining parts are the motor cortex and the somatosensory cortex. So the motor cortex is part of the frontal lobe, and the somatosensory cortex is part of the parietal lobe. They have similar functions. So the somatosensory cortex is for touch, and we know a lot about how different parts of the body are localized to different parts of the somatosensory cortex. And then the motor cortex is for voluntary behavior. So there's a part of the motor cortex for the lips, for the hands, for the feet, etc. And this is what initiates commands for the body to move. Kind of makes sense that it would be near the frontal lobe. The frontal lobe is the decider, if you like. You guys are probably too young to remember George Bush, but former president of the United States, and he would say, "I'm the decider." So the frontal lobe is the decider. And then it can send signals to the motor cortex, which then sends signals to the body. Importantly, so here's a representation of how different parts of so the blue is the somatosensory cortex, one half of it, and this is the motor cortex, one half of it. And it's not proportionate to the size of those body parts, which should make sense. So take touch as an example. My fingers can detect subtle differences in texture in a way that my knee can't. So there's obviously more nerve endings in my fingertips than in my knee. And the same is true for movement. I can do subtle, types of movement with my hands, whereas my knee, basically I can do this, and that's pretty much it. If the human body, and this is a little bit gross to look at, but if the size of our body parts corresponded to how much space they took up in the motor and sensory cortex, this is what we would look like. So, if you got set up on a blind date with someone who looked like that, you'd not be very happy with your friend. Right? So obviously, lips and the hands are much larger. So that's the function of those two. And actually I have a video of one of the ways we initially localized what different parts of the cortex did, which is Wilder Penfield, a Canadian neurosurgeon. While people were on the operating table, he would stimulate different parts of the brain and look at what they reported. This is completely painless. The brain doesn't have pain detectors, and also perfectly safe. And so you'll see if you stimulate one part of the brain, like let's say a part of the sensory cortex, people will report feeling like a tickle in their toe. You say, okay, that's the toe part. So see some footage of that. Now, if video of an open brain makes you uncomfortable, you can close your eyes. Okay, so one little question I have for you all. This will help you remember what these four things do. I'll make it a bit more, I'll give you all some skin in the game. Imagine that an evil genie came to you and said, I'm going to destroy one of the four lobes, but I'm going to let you choose which one. So just to quickly remind you all, so the occipital lobe is the early stages of conscious visual processing. So if that's gone, you're blind, right? At least consciously. Temporal lobe is the primary auditory cortex, so you're going to be deaf, probably going to be a little confused visually, and you'll also lose quite a bit of language comprehension, because a lot of language processing is in the temporal lobe. If you damage the parietal lobe, you're going to be numb and bad at spatial analysis, so no sense of touch and poor spatial reasoning. And then the frontal cortex, you're going to be unable to move, and at least very much, and unable to think or have difficulty with complex processing and impulse control. So the kind of funny way I summarize it is you can either be blind, deaf, numb, or dumb. It's kind of a choice. So think about the four. Any questions about the function of the four before we take a poll? So write down which one you would ask the genie to damage. And pretend he says, I'm going to kill you if you choose nothing. So you can't choose nothing. And the four lobes might be important on the test. Nudge, nudge, hint, hint. So, show of hands, who picks occipital? 1. Who picks temporal? 4. Who picks parietal? A bit more, 6, 7. Who picks frontal? Okay, many of you did not vote. I'll let you think about it at least. So that's all I have to say about the four lobes. Any questions? Okay, I'll keep moving through some other terms. So the next term is mirror neurons. There are neurons that are designed to respond both when you do something and when you watch someone else do something. So as I do this silly motion, my motor cortex is lighting up, but so is yours, same part of the brain. So the neurons that fire both when you do this and when you watch someone else do it are mirror neurons. These are important for observational learning, so learning from watching someone else do something, and for empathy. So that's what mirror neurons do. Another important term is neuroplasticity. So, related term, we would say the brain is highly plastic, and plastic here doesn't refer to the material. Plasticity means the brain is flexible and can change in response to damage or new experiences. Plasticity is highest when you're young. The brain, when you're young, is highly plastic, highly flexible. It can grow new connections and adapt in that way. Just to give you a sense of how extreme this is, there are a handful of crazy cases where kids have a life-threatening condition that requires that an entire half of the brain be removed. And you might think, wow, those kids must be fucked. You lose a whole half of the brain. What hope is there? But in fact, if they're young enough, they will likely show hardly any deficits when they grow up. The other half just grows new connections and kind of makes up for what's lost. There's a really funny psych podcast, especially the older episodes, called Very Bad Wizards. The psychologist on the podcast, David Pizarro, his niece, he talks about it on the podcast, his niece had this done. So a whole half of her brain removed. She's now an adult, in college, normal range IQ. The only remaining symptom is on one half of her body, she walks kind of awkwardly. But other than that, you probably wouldn't notice. Now, if you removed half of my brain, yes, I would be fucked. So plasticity is highest when you're young. So just so you know how this works, when the kid is born, they have basically all of the neurons they will ever have. What happens as they develop is the neurons grow more connections than they need and lose whatever connections don't get used as much. So it's a process of growth and pruning. Some have likened this to the process of evolution. There's a term you'll see in the literature, neural Darwinism. So it's kind of similar to evolution, right? There's lots of random genetic variation, and then some of those lines die out. So too with the brain. It grows more connections than it needs and loses the ones that don't get used. There's then a section on a growing field now, which might sound like an oxymoron, which is cultural neuroscience. So, growing up in a different culture can shape how your brain reacts to things. One of my favorite examples of this, I took a cultural psych course here at Douglas. There are parts of the brain that light up when you're thinking about yourself, at least in individualistic cultures. The same parts of the brain, if you grow up in a more collectivist culture, they light up both when you're thinking about yourself and when you're thinking about people very close to you, which is consistent with the idea that they have a more interdependent sense of self. They don't view themselves as atomized individuals, they view themselves relationally. So, I don't know as much about this area, but that's an example of the kind of study done by a cultural neuroscientist. Okay, the next section is on genetics, epigenetics, and associated topics. We're just going to skip that. This book has, or the loose leaf at least, has a chapter from a different textbook slapped on at the end. Next week we'll be covering that. So it goes into those same topics, but in more detail. So the next section of the book is on different ways of studying the brain. The first one we've already seen a bit of, which is brain damage. One convenient way of studying the brain is just to look at cases where people have parts of their brain damaged, and you look at what they can't do anymore. So we already saw Henry Molaison. Another case, probably the most famous brain damage case, is Phineas Gage. How many of you have heard the name Phineas Gage? Handful. He's the guy who had the iron rod go through his eye, so some of you may remember that. So he was a railroad worker in the 1800s. Explosion went off, sent a rod through his brain, damaged his frontal lobe, and he had various personality changes result from this. So we'll see a video of the reenactment. Again, they do reenact that event, so close your eyes if you're squeamish about that sort of thing. Okay, now I should note, for the sake of completeness, there is controversy about the extent of Gage's personality change following that incident. We don't have actual footage of it, so some reports differ. However, the fact that the frontal lobes are important for self-control and regulating the impulses coming from the limbic system, that's very well replicated. A different case of brain damage, well, I guess brain surgery in this case, that is also well replicated is the role of the corpus callosum. So here we have to talk about the two hemispheres. So your brain's divided into two hemispheres, the left and the right. And there's a few things you have to know. First of all, control of the body is contralateral. So each hemisphere controls the opposite side of the body. So the right motor cortex sends commands to the left side of the body, the left motor cortex sends commands to the right side of the body. So you have to know, and I should say too, for vision, that's also the case. So information from the right side of the visual field goes to the left hemisphere, and information from the left visual field goes to the right hemisphere. And some people think, oh, that's like, goes from my left eye to the, but it's not about the eyes. If you close one eye, you can still see some shit over here. So it's, it has to do with the left and right visual field. That's what gets reversed. So you have to know that. There are also two parts of the left hemisphere you have to know about. Broca's area and Wernicke's area. If you're in my 1200 class, you've already heard about these. So these are both in the left hemisphere. I'll actually bring up a picture of this. So the left hemisphere is dominant for language use. So, which makes sense, language is a very important function for humans, and if you're right-handed, the left hemisphere is the dominant hemisphere. So for the vast majority of right-handed people, language is localized to the left. Left-handed people are all mixed up. Some of them it's more equal, some of them it's dominant in the right, some in the left. But for right-handed people, the vast majority of the time, language is localized to the left. So I'll start with Broca's area. Broca's area is in the left frontal lobe. It's near the motor cortex, and it's important for language production, speaking, writing, etc. So it's near the motor cortex, so the motor cortex is what sends signals to the lips and tongue to speak, so it makes sense that Broca's area would be nearby. So, as is always the case, we look, not always, but often the case, we learned about this through brain damage. So the neuroscientist Paul Broca studied a case of someone who had damage to this area, and he could understand language. If you gave him a complex verbal command, go downstairs, open the drawer, and get this thing from the back, he can do it. And he can make noise, he can make noise with his face, but just can't produce sentences, can't produce language. Wernicke's area is important for language comprehension. So it's near the auditory cortex, which is for hearing, and damage to Wernicke's area leads to impairments in language comprehension. So they can speak language, but it doesn't make sense. They utter a bunch of nonsense if they have that part of the brain damaged. So like words, but not in a sensible order. Okay, so that's background. Now, here's where the corpus callosum comes in. The two hemispheres are largely independent, but there's a set of axons called the corpus callosum in the middle that allows for each hemisphere to send signals to the other one. So that way they can keep informed on what the other one is doing. One treatment for, yes?
Is corpus callosum one word or 2 words?
2 words. Corpus callosum. It should be on the key terms list. So, one treatment for epilepsy that was more promising than the Henry Molaison strategy is cutting the corpus callosum, thus making the two hemispheres almost entirely independent. So they can no longer send signals to each other. Now, believe it or not, from the inside, they report experiencing no change. And if you watch them, except maybe for the first few weeks, you notice no change. Immediately after the surgery, they'll sometimes do goofy things like, one hand will turn a page and the other will turn it back, and they'll fight over the page, but that tends to go away. But if you bring them into the lab, you can elicit really interesting responses. So one thing they get them to do, and I'll show footage of this, you get them to look straight ahead at a fixation point, and then you can flash different images to the two hemispheres. So if they're looking straight ahead, you can flash one image here, only the left hemisphere, quote, sees it. You flash another image over here, only the right hemisphere sees it. But remember, the left hemisphere is dominant for language, so they can only talk about it if it gets flashed to the left hemisphere. So you flash an image to the left, they're like, oh, you showed me an apple. Flash something over here, what'd you see? Oh, I don't know. But they can still draw it with the hand controlled by the right hemisphere. If you tell them draw what you just saw, just take a guess, they'll draw the thing they saw. So I'll show some footage of one of these cases. So these people are called split brain patients. So in that case, when he's asked, why did you draw the saw? He says, I don't know. In other cases, split brain patients will engage in confabulation. So they'll make up a reason. And it's not that they're lying, it's that we have this habit of making our behavior sensible to other people. So they'll say things like, oh, it goes good with the hammer, so that's why I drew it. And you might think, wow, that's really absurd. But normal people do this too. There's a famous experiment in psychology where they had 5 identical items of clothing. I think in the original it was pantyhose or something. Whatever, it could be 5 scarves, 5 identical items of clothing. And people are asked, which one is your favorite? And they'll feel around, and much more than chance, they pick the one on the right. People just have a bias towards picking the one on the right. And you ask people, why did they choose it? They're not aware of this bias, and they'll make stuff up. They'll go, oh, it's got a really good texture to it, or I like the weight of this one. And they show all sign of being perfectly honest, but they're confabulating. They're making up a reason, or their brain is making up a reason why they did it. There are some funny advantages that these split brain patients have. We're all going to do a little task that you will all find very difficult, but someone with the corpus callosum severed, this is very easy. Take 2 writing implements, pen, pencil, at the same time, draw a circle with one hand and a square with the other. No pauses, just continuous drawing. So give it a few tries, see how you do.
Are we allowed to look?
Sorry?
Are we allowed to look, or are we looking at?
Sure, you can look, yeah.
This is hard.
So yes, so that's the point of this. Yes, it is hard. One person in my other class said they could do it okay, but you can try when you get home, see if you can get better at it. But give this to a split brain patient, no problem. They can do it first try. So not a huge advantage, but it is an advantage there.
Does it also work for like cabinet theory?
Yeah, things like doing things independently with the 2 hands is easier for split brain patients. Okay, last section of the chapter is on different brain scanning techniques. I have a couple of videos of 2 of them. You can, broadly speaking, divide the brain scanning techniques into 2 buckets. And finish your conversations after class. Those that give you a video, so a video of the brain's functioning, and those that give you just a picture. The first one that sort of gives you a video is EEG, or electroencephalograph. This is one that I used at the University of Alberta. I worked in a neuroscience lab that had an EEG machine. This one involves putting a cap on someone's head, typically with electrodes attached. What it measures is electrical activity at the scalp. I didn't know this before joining the lab, but you also have to put gel underneath the electrodes. You have to kind of brush their hair out of the way and put gel in. Some of them you soak the whole thing, but you need something like that to make the connection better. This technique has pros and cons. What it has is very good temporal precision, meaning time precision. You can know exactly when there's a change in activity. Typically, it's used to extract brain waves, so you can see brain waves. So if you see in a movie someone with electrodes on their head and there's brain waves on a screen, that's EEG. But because it's electric, it's basically instantaneous. What it lacks is spatial precision. It's not very good at knowing exactly where there's a change in activity because it's only measuring the scalp. So we just know there's an activity somewhere near this electrode. If you want to infer about activity in deeper regions of the brain, there are statistical techniques you can do, but they're more, less well established. So good time precision, poor spatial precision. There are two picture techniques you should know. Many of you have probably had these done at one point or another. There's a CT scan, which is just X-rays but for the brain. And an MRI, which also gives you a picture, a 3D picture. It sends a magnetic pulse through the brain. And we'll see a video of a neuroscientist explaining that in more detail, but this also allows you to get a 3D picture. These are less important for psychology because we're interested in moment-by-moment changes in brain activity. These would be used if someone had brain damage and we wanted to know which part of the brain is affected. Of course, they're also used in fact, more often used in medicine to identify tumors and stuff like that. The most commonly used technique in psychology research is fMRI. I don't know why the F is lowercase, but for some reason it is. So the F stands for functional. This involves getting a series of MRI 3D pictures. And what it can measure is changes in blood flow, in oxygenated blood flow. The idea is if one part of the brain is active, it gets tired and more blood flows to that part of the brain. So you can then make inferences about which parts of the brain are more active during different psychological tasks. This has the opposite profile of EEG. So EEG, I said, has good time precision, but poor spatial precision. This has the opposite. So spatially, you get a 3D video of the brain's activity, but there's two reasons it has bad time precision. One is the blood only flows to that part of the brain after it's active, maybe a second or two after. So this part of the brain gets tired and more blood goes there. The other reason is you can only take a picture every second or so, maybe two, I forget the exact amount of time, but you can't take it boom, 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 boom. So there's a bit of a delay leading to even less time precision. So I'll show two videos, one of EEG in action and one of a neuroscientist explaining fMRI, and then I'll close with one final technique. Okay, and then the last term of the day is transcranial magnetic stimulation. So all of the techniques we've just been talking about are by definition correlational. You can only correlate activity in different parts of the brain with different psychological functions. Transcranial magnetic stimulation allows you to stimulate different parts of the brain, either activating or deactivating them temporarily, and you can look at how that affects responses. This was done with animals for a long time and shown to be completely safe. So now it's used a lot. I actually knew a grad student at Georgia State who got to use this. I was very jealous. just as an example, you can stimulate parts of the frontal lobes involved in complex reasoning and change people's responses to moral dilemmas. That's the kind of thing that's done. Any questions on the neuroscience chapter? Okay, see you all next week for genetics and evolutionary psychology.